It's my pleasure to lead to the stage Yvonne Vujkovic-Cvijin, who is going to talk to us about association versus causation, confounders, transcending confounders in microbiota research. Yvonne, you can go on camera. There you go. And if you would share your screen with us. Excellent. Awesome. You are good to go.
Great. So you can see my screen?
We can.
Awesome. Thank you so much, Len. And thank you for the heroic pronunciation of my name. It's obviously not easy. But yeah, so I'm Yvonne Vujkovic-Cvijin, and it's been a pleasure seeing some of these talks. And thank you so much for inviting me to share these data with you. I hope that they're informative to some members of the audience. Okay, so as many of you know, the microbiota is a critical organ, and many studies, a mountain of evidence at this point has shown that several host extrinsic and host intrinsic factors like genetics can alter the microbiota to create a microbiota compositional state that can contribute causally to aberrant and elevated inflammatory levels that can then spur pathology in a number of immune-mediated diseases like IBD and NASH. And now it's very important to state that the majority of studies and pieces of evidence that support this causal chain of events have been derived from animal models. And the field is really yet to understand whether the microbiota is truly causally related to these diseases in humans, and to identify specific microbiota constituents that might be driving differential outcomes in these diseases. And I think that part of the reason why we haven't yet been able to do so in humans is because we have a limited toolkit in microbiome science. And importantly, the tools that we do have at our disposal have a number of caveats that, if explored further, I think we can potentially overcome. And just as a brief introduction to some of those concepts, to the caveats of the tools that we have, murine studies, of course, are extremely useful. And one distinct strength of them is that we can perform what some call precision microbiome editing. And I mean that in the sense that we can give to mice a bacterium or fungus, a microbiota constituent that we think is deleterious and contributing to disease, and examine its effects on pathology, and we can do the same with bugs that we think might be protective. Now, an important caveat to the study of mice and their relationship with the microbiome is what we call the species barrier, and that's embodied well in the observation that SFB, which is among the most immunomodulatory gut microbiota constituents in mice, when given to rats, is immunologically inert and has no effect on the immune system of rats, in contrast to mice. And that really suggests that when we discover, pathogenic or protective species in mice, they may not have the same effect in humans. And when we take human gut microbes and put them in the mice, that experiment may not recapitulate their behavior in the original human host. Now, of course, there are some human studies there are some tools we can use in humans to study them. And one advantage of studying humans is, of course, that the model now is the target species. And one of the primary workhorses to this point of human microbiota research has been the cross-sectional association study, where we take disease subject and healthy individuals and compare their microbiomes. But a very important drawback to this kind of study is that the conclusions can be confounded. And by that, I mean the following. So I'm going to illustrate that concept just in a very simplistic hypothetical study. So in this hypothetical case control cohort, we see that individuals that consume an enormous number of calories seem to be leaner than subjects that consume comparatively fewer calories. And this would produce the spurious inference that consuming more calories makes you leaner. And of course, that's likely untrue. And if we look closer at the underlying characteristics of this hypothetical study, we find that the individuals that consume enormous amounts of calories are actually marathon runners. And we were comparing that to the general population. And in this case, of course, these two groups were confounded by energy expenditure And if we were to match our cases and controls by energy expenditure, we would likely see the true association, which is that more calories leads to higher BMI. Now, this may be a somewhat obvious mistake that one might make, and therefore something that we can easily avoid in human studies, but there are more insidious ways that confounding variables can creep into our study designs in microbiome science. And one salient example of that is in the space of the HIV-associated microbiome. And just as a brief background to the study of the microbiome and HIV, we, from decades of research, have built a large body of evidence to suggest that individuals, even after antiretroviral therapy, have elevated levels of immune activation, and that the extent to which the immune system is activated in these subjects is really tightly correlated with the chance of early death. So even despite almost undetectable viral loads in blood, these individuals seem to be dying early, and it's linked to elevated levels of inflammation. And because it seems that the virus may not be involved anymore in that elevated inflammation because it's nearly undetectable in blood, some folks, including we and others, have postulated that the gut microbiome may play a role in that inflammation. And we found that certain patterns of microbial composition, gut microbial composition, correlated with some of those most predictive markers of immune activation. And since our study, many investigators came out and found similar shifts in microbial composition in the gut microbiota of HIV-infected individuals. But importantly, a few seminal studies found that actually sexual behavior and men who have sex with men have a very different gut microbiota composition than men who have sex with women. And this is particularly relevant in the study of HIV because in the Western world, the predominant population of people with HIV are men who have sex with men. And so if we were to randomly select seronegative controls that are matched for BMI and age and sex, but we're not necessarily matched for sexual behavior, they would likely be mismatched for sexual behavior because among seronegative controls, of course, the majority of individuals are non-MSM. And so that's a problem if the MSM microbiota itself is unique from that of non-MSM, because perhaps when we compare people with HIV to seronegative controls, that we were really obtaining the signal associated with sexual behavior and not that of HIV. And indeed, if you look at some of these studies, while there were several studies were matched for sex and sexual preference, a majority of them were actually not matched for MSM status. And looking closely, the salient features of the MSM microbiota crept up into what was purported to be the signal associated with HIV infection in the form of lower Bacteroides and higher Prevotella. And this really sent the field reeling and begged the question, does the microbiota really differ in people with HIV, or was everything we were seeing related to this confounding variable of sexual behavior? And ultimately, if there are differences, are they at all relevant clinically? Are they linked with this inflammation that leads to early morbidity and mortality? And so we embarked on a study to try and address those questions, and we recruited a cohort of men who have sex with men, some of which were people with HIV and some were seronegative. And we see when stratifying by sex and sexual behavior, there is still a difference that we see in that's dependent on HIV infection status. And here are the centroids of the two respective groups, people with HIV and seronegative controls. And when we look at women, or when we look just within men who have sex with women, we see the same thing, that the HIV infection status segregates individuals by their microbiome profiles. So the HIV status pervades that of the confounding variable in terms of effects on the microbiota. And then we collapse these differences in the microbiota that are dependent on HIV infection into a single variable. And we found that individuals with a high prevalence of inflammatory comorbidities had a more perturbed microbiota than individuals with no inflammatory comorbidities, suggesting that perhaps the shifts in the microbiota are clinically relevant, though of course we need to do much more work to understand that and the causal relationship between the two phenomena. But in the course of this work, we came across something that I found very disturbing, which can be seen in these two graphs. So in the graph to the right, the colored dots above this dotted line are the taxa that have significantly differential abundance as a function of HIV status. So bugs that are either highly enriched or depleted in people with HIV compared to seronegative controls. And if we look here, these are the taxa that differentiated individuals by their sexual behavior. So MSM versus men who have sex with women. And just looking at the sheer number of dots, there's a huge difference in that there's far more taxa that are modulated by this confounding variable that we initially were not necessarily interested in, as compared to the disease variable that we were interested in, that the study was designed to capture. And this told us that this confounding variable, was much stronger than the actual variable of interest. And importantly, we, at the beginning of this field of study of the microbiome and HIV, we had no idea that this was a confounding variable, that sexual behavior would have such an impact on the microbiome. And it really made me think, it made us think, wonder whether there are other host variables that may similarly be confounding our studies, adding huge lists of false positives to our disease associations that we might not even be aware of. And so to try and rigorously address that question, we examined prior literature to see, first of all, what are people matching for, if for anything. And we actually found that in even high impact, nature cell science, high impact studies, matching cases and controls for confounding variables was sporadically done, and sometimes not at all. And importantly, there seemed to be no consensus on what variables to match for. Each study seemed to match for different variables. And so yeah, we wanted to rigorously identify potential confounding variables. And to do that, we developed a machine learning analysis framework, leveraging that with the American Gut Project, which is at the time, and I think still is, the largest publicly available data set of gut microbiome profiles. And importantly, they had also administered 124 question questionnaire to these individuals that spanned dietary, lifestyle variables, physiological variables, and disease statuses for these individuals. And this was done in collaboration with a very talented young bioinformatician, Jack Sklar. And together we developed this machine learning framework to essentially measure the strength of association of each of those variables with underlying microbiome profiles. And for sake of time, I won't go into details, except to say that for each binary questionnaire variable, we obtained what are called AUROCs that are essentially proportional to the strength of the robustness of the microbiome patterns in segregating individuals by a given variable. So proportional to essentially how strongly that variable has an impact on the gut microbiota. And just to show you what our results looked like in a brief example, so here, each of these categories was compared to individuals who reported never consuming meat or eggs. And we see individuals that report daily consumption of meat or eggs have a high AUROC to the right on the x-axis, indicating a high difference, a highly reproducible difference in their microbiota compared to never consumers of meat or eggs. And there's a dose-dependent decrease of the strength of that effect that was proportional to a decrease in frequency of consumption of meat or eggs. And these are some of the strongest dietary variables that we found had an impact on the microbiota. It included salted snacks, dairy, alcohol, and others. And we also found other host-associated variables having a strong impact on the microbiota, like sex, BMI, and bowel movement quality and frequency as well. And when we collapse all these AUROCs, these strengths of association with the microbiome of each variable into categories of diet, lifestyle, physiology, and disease, we find something that's very reminiscent of what I showed you before, which is that the disease itself, the disease variables, seem to have much lower strengths of association with microbiota profiles than these other non-disease related variables. So lifestyle, diet, and physiological characteristics, very reminiscent of the impacts of HIV in the microbiome versus that of the confounding variable of sexual behavior. And we wanted to delve more specifically into that observation, and we first were most interested in the top hit of our diseases that had a strong impact on the microbiome, and that was type 2 diabetes. We were especially interested in type 2 diabetes because there was quite a bit of controversy in the microbiome and diabetes field. Many studies had come out showing a strong impact, a strong association between gut microbes and diabetes, but each study seemed to find different and non-concordant results. And so we wanted to look closely into diabetes. And what we found was that when we take our diabetic subjects in the American Gut Project and randomly select healthy non-diabetic controls from the same cohort, that the controls in diabetics differed substantially in their distribution of alcohol intake frequency. And the same was true for BMI and age. Diabetics tended to be to have higher BMI and to be older. And accordingly, we saw very strong differences in the gut microbiota profiles between diabetics and control subjects, which with again the centroids indicating the group microbiota profiles, and they segregated quite robustly. Now, I showed you earlier that these variables had a strong impact on the microbiota, and so we wondered how much of the differences that we're seeing here between diabetics and controls are really actually due to these confounding variables as opposed to disease itself. And so what we did was we went back and we reselected healthy control subjects that had the same distributions of these variables. So individuals that were matched for alcohol intake, BMI, and age. And we saw that there was a dramatic reduction in the microbiota differences between diabetics and control subjects when matching for these important variables. And now you may ask, how realistic is this, what we engineered up here, the comparison that we made here? Do studies in the wild really, is it realistic that we might see this level of mismatching among studies out there? And we were surprised to see that actually, even among high impact studies, that none of them actually matched for all three of these confounding variables, likely because alcohol intake was an underappreciated confounding variable. But what was interesting is that each study seemed to match for different confounding variables, and which made us think that perhaps the discordant results that were reported among these studies may have been due to the studies being confounded by different variables, essentially reporting the effects of different confounding variables. And we next wanted to understand, because there were a host of diseases that were captured in the American Gut Project, we wanted to understand whether diabetes, the observation we made for diabetes, was unique to that disease, and so looked at whether selecting healthy controls for each disease in the AGP might produce similar confounding effects. And we saw that for the vast majority of diseases, when selecting healthy controls randomly, there was significant differences in the distributions of these various confounding variables. And when we look at the gut microbiome differences between cases and controls in these diseases, we see that for most diseases, when we have randomly selected controls, there are statistically significant differences between the cases and controls. And to the right of this dotted line indicates significant differences between cases and controls in terms of the gut microbiome. But then when we do something similar as what we did with type 2 diabetes and go and reselect healthy controls that are matched for these confounding variables, we see a dramatic reduction in those significances in terms of the differences between cases and controls for nearly all of the diseases. And some of the diseases now no longer become significant in terms of their association with the microbiome, a lot of these neurological diseases. But importantly, we were intrigued to find that IBD, even when controlling for confounding variables, has a robust signal in terms of the microbiome. But at the end of the day, even even though we think that this methodology of controlling for confounders can really help us reduce the number of false positives that we discover in our association studies, and thereby help us focus resources for understanding mechanisms between that govern the relationship between host and microbe for the most relevant bugs. At the end of the day, even after matching, an association study still doesn't tell us the cause and effect relationship. Do these microbes, for example, in an inflammatory disease like IBD, causally contribute towards inflammation that drives disease? And we really, thought about that question a great deal of how do we move forward from association studies to identify causal contributors to immune activation. And several groups have sought to address that question, and they've really looked to the B cell arm of the adaptive immune system. So B cells secrete immunoglobulins that are often specific and highly targeted to an invading microbe. And importantly, they're highly compartmentalized in terms of their secretion, induction and secretion, such that IgA, for example, is predominantly expressed at mucosal surfaces. And groups have found that gut commensals that are highly targeted by IgA can contribute to inflammation at the tissue from which they were derived. But subsequent groups have also found that the vast majority of bugs that are simply residents of the small intestines can elicit IgA. And so relying on IgA as a marker to tell us what bugs might be pro-inflammatory may have the drawback of having, again, false positives, because even non-pathogenic, non-inflammatory commensal species can induce IgA against them. And so we became interested in IgG, which is a comparatively less studied immunoglobulin in terms of its relationship to the microbiota. And we were interested because it is really predominantly mounted against invasive pathogens, bacteria that have reached the gut barrier and are associated with inflammation. And we know that in IBD, it's been shown for many years now that subjects have higher IgG targeting of gut bacteria than do healthy non-IBD controls. The only question is what are the identities of these gut bacteria targeted by IgG? And that's been really poorly understood. And so we sought to create a framework to identify IgG targeted bacteria by taking fecal samples and paired serum where the IgG is found, co-incubating them to allow for the IgG to bind the gut microbes in the feces, and then separating the IgG bound bugs from those that are unbound, subjecting them to 16S sequencing, and calculating this IgG score that's proportional to the titer of IgG against each gut bacterium. And in this way, we can quantify IgG titers to essentially all detectable gut bacteria that are in the sample. And we wanted to understand whether this is truly identifying bugs that have crossed the gut barrier. And so we employed this model of gut barrier disruption that involves infection with the parasite Toxoplasma gondii. And this causes small intestinal barrier breach. And so we treated mice with this parasite and then harvested their spleens and livers and cultured those organs in order to identify what bacteria had crossed from the gut and entered those organs. And then to a similarly but separate group of mice, similarly treated but separate group of mice, we infected them with toxo to cause that barrier breach. And then we allowed the immunoglobulin repertoire to form and performed IgA and IgG-seq to quantify the IgA and IgG titers against all gut bugs. And we found that when we compare IgG scores in toxo-infected mice to uninfected mice, the bugs that translocate reproducibly, shown in these colors, seem to have very different IgG scores in infected mice versus uninfected mice. So as a function of barrier breach. Whereas if we look at IgA, IgA was not able to identify the bugs that had translocated like IgG was. So we find heightened IgG against bugs that had translocated. And we wanted to understand whether this might be occurring in humans as well. And so we worked with Suzanne Devkota and her postdoc, Connie Ha, here at Cedars, who did an amazing study where they recruited a number of IBD subjects who were about to undergo bowel surgery, so resection of the bowel as part of manifestations of disease, as a treatment for severe manifestations of disease. And what they did was they took mesenteric adipose tissue from these surgical resections and cultured it to identify bacteria that had crossed the gut barrier and entered this mesenteric adipose compartment. And so they identified bacteria that had translocated in about 29 IBD subjects. And so what we did was we applied our IgG-seq approach to understand whether in the individuals that were translocation positive for a given bacterium, did they exhibit higher IgG scores against that bacterium than subjects that were translocation negative. And indeed, that is what we found. We think that this supports the idea that IgG-seq, that methods to quantify the systemic IgG repertoire against gut bugs can help us identify bugs that have translocated and are therefore potentially involved in activating the immune system and therefore potentially involved in driving inflammatory diseases. And as part of that study, we were able to compare the total IgG repertoires of healthy subjects to IBD subjects. And right off the bat, we saw that that Crohn's and colitis subjects had much higher IgG targeting against gut bugs than did healthy individuals. And this was consistent with many prior studies. But what we could do that prior studies were not able to do yet is really look at the individual taxa that are targeted by those systemic IgG repertoires. And when we take a holistic approach, similar to a PICO-A plot and two-dimensional microbiome analyses, and look at the similarities of microbiome anti-microbiota repertoires against all gut taxa, we see that the Crohn's and colitis subjects really cluster away from the healthy subjects by their anti-microbiota IgG repertoires. And when we look at specific bugs that differentiated Crohn's and colitis from non-IBD subjects, we find that a number of intriguing taxa seem to be targeted by IgG much more in IBD than non-IBD subjects, including Collinzella, which was among the only taxa that predicted severity of disease in a large cohort of pediatric Crohn's disease subjects. So we're very intrigued to find that. And we also were very intrigued to find Bifidobacterium. These two genera are largely underappreciated and understudied in terms of their role in IBD. So we were surprised to find them being targeted, but very intrigued, of course, and we're going to follow up on these observations. But we find that when looking across countries, using publicly available metagenomics data sets, B. longum, Bifidobacterium longum, correlates very strongly with IBD prevalence across about 47 countries. So we're very interested in that and hope to follow up on that. And just to briefly summarize what I've shared with you, we think that we've presented evidence that host physiological, dietary, and lifestyle variables have a very strong effect on the gut microbiota, and that this can obscure relationships between the gut microbiota and disease. And we think that the field will likely gain clarity on in terms of identifying bugs that are truly associated with disease if we match our comparison groups for these variables that we identified. And this may be useful for target discovery, for subgroup classification within disease, and also for treatment prediction. If we allow these variables to insert the noise that they do in our studies, we may be more limited in terms of achieving our study goals. And I've also shown data that seem to suggest that antimicrobiota systemic IgG can identify translocating pro-inflammatory gut microbial taxa. And in work that that's in the early days, that's ongoing, we're very interested in something that I think is relevant for this audience. We're very interested in using Bayesian inference and combinatorial testing strategies in vivo to interrogate the effects of dietary supplements on the gut microbiota. And we're really optimistic that this can unlock precision microbiome editing strategies that are safe in humans. I think most of you would agree And we're very interested in this space. And if anyone would like to collaborate and potentially provide us with pure, reproducibly sourced dietary supplements or food compounds for us to test in our Bayesian networks framework, please reach out to me. I would love to chat with you. And also, we're looking for postdocs. So if anyone's interested, please let me know. But as a final, just to acknowledge all the folks that contributed greatly to these studies. So collaborators at UCSD, UCSF at University of Amsterdam. Here we're at Cedars-Sinai, and these are folks in my lab and all our very important collaborators, as well as collaborators at NIH and my former mentor, Yasmin Belkade. Thank you very much for for your attention and for allowing me to
Thanks very much, Yvonne. I think some subject matter that needs a lot more digging into. We're a little bit pressed for time. We do have a couple questions, and I think there's a couple things that I want to follow up and maybe we have a follow-up interview conversation. Also, we operate the Global Prebiotics Association, so I think that we'll have some folks that may reach out. There's also a Young Researcher Award that's being offered by the GPA. That may be something of an incentive for us to connect with. But let me get to the questions first. Obviously, not enough people are talking about confounders. What's your guidance for reanalysis of previously conducted research or new research that's at the right stage for this interpretation, analysis, and the identification of these variables? Maybe we parse that out to what's the re-examination and what's the new examination?
Yeah, thank you so much for that question. Yeah, so we actually, as part of our paper, it's in there, it's a supplemental figure. We did go back to some studies and reanalyze them. Particularly, we focused on type 2 diabetes, but we also looked at metabolic syndrome. And we did find that when you reselect healthy controls from within prior studies, that you do get a reduction, similarly, a reduction of the taxa that differentiate cases and controls. Presumably getting rid of some of those false positives. And actually, we do show that some of the taxa that now drop out in terms of being significant are taxa that are associated with the confounding variables. So there's definitely potential value in going back to some studies that did collect information on some of these confounding variables and reanalyzing them. So I think that's great. It's not that we can't gain from what's in the past. And what's been done in the past also does include the bugs that are truly associated with disease. It's just it has also likely a lot of these confounder associated bugs. But of course, not all studies collected information on alcohol intake and bowel movement quality and things like that. And so there's a role, I think that there's the value also to new studies reevaluating these associations that we've found before in the context of being able to match and account for these confounding variables.
Do you imagine an exhaustive list that's subject to additions of all these variables that people have found and their interferences or their confounder net impacts? Is this a data set that ultimately gets created?
Yeah, so I mean, we provide something along those lines in our paper as part of like supplemental data. But I think that, because there's such vast differences in due to batch effects, I think that we cannot always apply the lists that we get of taxa that are associated with these confounders to other studies. Like we can't port that reliably because of DNA extraction differences and also, metagenomics versus 16S. Those are difficult to cross compare. And also the limit of detection. We had to rarefy down to something like I think 8,000 sequences, and so we're not getting all of the rare taxa that might be associated with these confounders that might be very important in disease or that confounding variable. And so I think that what the image that any one study can give might be incomplete and not transferable to others. So that's why we do encourage that people match for these confounding variables within their own study to maximize chances of those not interfering.
All right. We do have a couple other questions and maybe we'll get to you post this and make sure we get them back to the people that did submit them. But I want to thank you, Yvonne, and close us out.